Okay. Thank you. I would like to thank the organizers for allowing me to talk to you about the improvements that we've made to PhenoMiner and to give you a little bit of a sneak preview for what's actually coming soon. Coming soon. I work for the Rat Genome Database, and we are the online resource for rat data pretty much worldwide. So just to give you a brief introduction to RGD's PhenoMiner. PhenoMiner is a tool for ontology-based storage and mining of quantitative phenotype data for the laboratory rat. PhenoMiner includes both data from high throughput phenotyping projects, which come to us relatively standardized, and scientific literature, which comes to us completely unstandardized. So to allow comparisons across studies, as well as flexible and intelligent querying, PhenoMiner uses ontologies to express what was measured in terms of the clinical measurement ontology, how it was measured using the measurement method ontology, under what conditions it was measured using the experimental condition ontology, and in what animals it was measured using the rat strain ontology. The vertebrate trait ontology is also used to group related measurements within a study, and PhenoMiner can be accessed through the Phenotypes and Models tab in the menu at the top of any RGD page, or using the button on the RGD's homepage. So thinking about some of the limitations of our first iteration of PhenoMiner, I started thinking about some of the comments that we have heard and some of the comments that we have made ourselves about PhenoMiner. And they're things like the process for selecting records to view is not intuitive. Once I've selected terms, it's hard to change my selections. I can only view results for one measurement at a time, even if I choose terms that use the same units. What? I can sort the results of that table? Oh, but when I sort the table, it doesn't change anything in the graph. That still appears to be ordered randomly. And coloring the bars in the graph by experimental condition is nice, but I'd like to choose what they're colored by. So to give you a little bit of an idea of what I'm talking about, this is the landing page of the original PhenoMiner, and it was designed to be flexible, and it is. But you come into this and you look at it and maybe you don't quite know exactly what it is that you need to do. So assuming that you figure it out, you select an ontology, and then you need to navigate through the tree, which means that you kind of have to know what the ontology is. And there is a search, but it basically takes you to the tree and makes you navigate through the tree. And then once you've decided and found the terms and chosen them, then it takes you to another page, which gives you further options of things to do. And that basically makes it so that you're going back and forth between the ontology tree selection pages and pages that ask you what you want to do next. In contrast to that, PhenoMiner 2.0, in this new iteration of the tool, all of the components are on a single page. So the strain ontology loads automatically when you first open it, but you can still start with any of the ontologies by using the tabs in the lower right panel. And as selections are made in the bottom panel, then they appear in the boxes above. So looking at those bottom panels, the lower left panel has a prominent search box, and then below that, an alphabetical list of all of the terms from that ontology that have data in PhenoMiner. So you can scroll through the list of terms, or you can enter a term in the search box. Once you enter a term in the search box, it narrows the list below it to only those things that match that search term. And then you have this, these select buttons, And if you choose a select button, it puts the term into the top box and also opens that, that region in the ontology tree in the bottom right panel to make it easier for you to refine the selection, to add more terms, and then also if you choose to take terms away. And if you do want to take terms away, then you can easily do that by either unchecking the box in that lower panel or clicking the X beside that term in the top box to just remove that from the search. Nope. Okay, there we go. Okay, so all of this, as I mentioned, all of the selections are made within the same screen, and all of the changes and additions and subtractions that you make are all shown in real time. And you can switch back and forth between the ontologies using the tabs in the bottom right panel. And at each step, options are limited by the selections that you've made. So if you've chosen some rat strains, the only options that you'll have for choosing clinical measurements will be terms that have been used with those strains in PhenoMiner, so you don't end up going down a path and then ending up at the very end of it with no data. So the options are limited by your previous selections. And as you go through, you can choose terms from any or all of the ontologies that are listed. If you choose not to make any selections for one of the ontologies, so for instance, if I choose rat strains and clinical measurements and experimental conditions, but I don't want to limit my options for measurement method, I don't have to make any choices for that, and it will show me all of the results for any of the measurement methods that match the other search terms that I've used. And so once you have your list of filters that you want to use, then you can just hit that generate report button and it will take you to the result page. So the PhenoMiner 2.0 result page consists of 3 parts: a graph, a list of filters, and a table of results. And you're looking at that and thinking, I see 2. I only see a list of filters and a table. And that's because if you choose measurements that use more than 1 unit, so in this case, I've chosen heart rate, which would be beats per minute, and several blood pressure terms, which would be millimeters of mercury. If you choose measurements that use more than 1 unit, it hides the graph until you use the filters on the left-hand side to filter that down to measurements that only use a single unit. So once I do that, in this case, I've chosen to only show my blood pressure measurements, then the graph appears. And the filters, the graph, and the table are all interconnected. So things that you do in one will affect all of the rest of them. You can select filters on the left to limit what's shown in the graph and the table. Selected filters are also shown above the graph as well as in the list to the left. And you can remove filters by either unchecking the boxes on the left or by clicking the X for that filter in at the top of the page. And I can choose any one or more of the filters, and the one on here on the left is you can remove all of the filters at once and it will take you back to your initial search results. So if I remove the measurements for mean arterial blood pressure, then it just shows the bars that correspond to systolic blood pressure and diastolic blood pressure. So one of the things that was requested was the ability to change the coloring in the graph. And so PhenoMiner 2.0 is more interactive, and it does include a color by function. The default is for it to be colored by experimental conditions, but you can change that and have it colored by strain or phenotype, method, or the sex of the animals. So if I change this particular graph and color it by phenotype, then I get this graph where I've got just the, I have just two colors, and there's a drop down legend that tells me that the yellow bars are diastolic blood pressure and the purple bars are systolic blood pressure. So the tool also allows you to sort the table, and sorting the table reorders the bars in the graph. And then you can use that kind of functionality. So for instance, using the color by phenotype and sort by value in this case, and it's easy to see at a glance the mix of diastolic and systolic blood pressure values. So one might have thought naively that all of the diastolic blood pressure values would all be on one side and all the systolic ones would be on the other side. But here it's easy to see that some of the systolic blood pressure values are actually lower than most of the diastolic blood pressure values, and some of the diastolic blood pressure values are actually higher than a number of the systolic blood pressure values. So if I go back to a graph that's actually looking at mean arterial blood pressure, where data for individual rats has been submitted to us, the graph shows not only the average, but also the individual values that have been submitted. So looking at this graph for mean arterial blood pressure, you can see that some of the bars include red dots that indicate the values for individual animals. So you get to see not only the average or the mean, you also get to see the distribution of the values that went into making that mean. So you can mouse over the bar and it will bring up a pop-up with details about the average value for that sample of rats under those conditions. And you can mouse over any of those dots to see the details for that individual rat measurement. So those values are also, you're also able to view those values in the table. If a row of the table has individual values, it will show the average value and also an option in the last column to view the individual values. So if you click the view values in that last column, it shows you the list of individual animal IDs and the individual measurement values that go with each of those. At present, the way the table is set up, you can't download all of the individual values, but that's high on our list of things to do to be able to not only see on the website, but also to be able to download not only the averages, but also the individual values so that you have the raw data that went into making those values. So all that to say that data from the query can be downloaded. At least the average values can be downloaded. A button for download all records is displayed on the pages for both unfiltered and filtered queries. And then for the filtered queries, there's also an option to download just the table view records. So just the records that you have filtered down to as you've played with the filters. So that is our soon to come new PhenoMiner tool. If you are interested in seeing it, if you want to stop me at lunch, I can actually show you the tool itself and kind of give you a run through. It's not out on the website yet, but it will be very soon. We're just doing final testing. We wanted to make sure that we tested it really well before we sent it out into the wild. But it's coming soon. And with that, I will say thank you. And I'd like to acknowledge the incredible team that we have of both scientists, scientific curators and developers who produce all of this stuff. Acknowledge our funders, the National Heart, Lung, and Blood Institute, and all of the researchers who contribute data and faithfully use our website and our data. And with that, I will take questions. Thank you. Thank you.
Question for you. When you were showing the right here. Hi,
sorry.
No, that's okay. When you were showing us the blood pressure data, I saw that there were 2 bars that had the exact same values, and they were listed as the same strain condition. Do individual studies have their own representation? How are you grouping things to make things easier to see?
Yes. I'm trying to make sure that I speak correctly. If the values come from different studies, they will show up as different measurements. Yes. And one of the things that you can see in the pop-up, but it's not quite as obvious in the graph, is you could have, if there was no difference between an animal that was fasted and an animal that wasn't fasted, they might show up as the same, with the same value, but they would be 2 different bars because they have 2 different conditions. So, thank you.
So I have a question. So, and I recognize that this is probably beyond the scope of what you're doing, but there are no, what I would commonly refer to, I guess, as phenotype calls. So there's no stats or saying, well, this rat under this condition has high blood pressure. For example, if we use your example, there were, bars of different heights. So is the expectation then, and you would need the individual data to do this, that users would then be able to download the data and run their own statistical analyses and things like that?
Yes, that's exactly the idea. And we do have qualitative curation that goes along with this. So in addition to the actual values, if there was a study that was done and they found that under these conditions, this particular rat was hypertensive, then we would have the qualitative phenotype measurement that says that it has increased blood pressure or it's has hypertension or whatever, in addition to the actual specific values.
Okay. And so what terms are you then using? So those phenotype calls that I guess you're mining from the literature and
Yes.
so relying on the peer-reviewed data and
Yes.
to make those calls. Are you using specific ontologies for those?
Yes.
And are they cross-mapped to, say, the mouse or human ontologies?
Yes.
In any way? Okay,
Yes.
great.
We use the mammalian phenotype ontology
Okay.
for the
Perfect.
rats. And those are also mapped to the human phenotype ontology, which, and we actually have data for human. So you would, in RGD, you would be able to look across the orthologs and say, rat has this phenotype, mouse has this phenotype, human has that phenotype, because they're not shared across the species, but we have the data for all of those species. So you'd be able to just within RGD be able to compare all of them.
Great, thank you. Any other questions?
Is there a plan to expand this? So, like the mouse site has gene expression and activity of Cree transgenic lines and things like that. Is that the future goal for this site as well?
Yes, absolutely. We're hoping now that we're getting a little bit more usable interface for the quantitative phenotypes. We want to be able to add in genotype data so that you can connect the phenotype, the specific phenotype and the genotype together and also the quantitative expression measurements, and be able to link, phenotype, expression, and genotype, be able to link all of those three together. So yes, that's the plan.
Okay, well, thank you so much. If you could join me in thanking all of the speakers from this morning. And enjoy your lunch.
No.
No. Oh, wait.